epigenomics. I'd like to introduce Dr. Bekim Sadikovic, who is at the London Health Sciences Research Chair in Clinical Genomics and Epigenomics at the London Health Centres in Ontario. He'll be talking about DNA methylation and epi-signatures. Thank you for coming, Dr. Sadikovic.
Thank you very much. I just got to figure out how to turn this thing on.
There's your expert right there.
Perfect. Hello, everyone. Hopefully everyone's caffeinated after the break. Thanks very much for having me here. Thanks to the organizing committee. I was just saying, I feel I learned more about the American regulatory and policy in genetics over the last 3 hours than I did in my lifetime. Now, I am in Canada, and I'd love to tell you that things are simpler and easier, more straightforward in a government-run genetics or healthcare system, and I'd be lying. They're just as complicated, so grass always seems greener on the other side. So thank you very much. The title of my talk today is DNA Methylation Epi-Signatures as Diagnostic Biomarkers for Rare Diseases and Beyond. I do have a recent disclosure. Some of the work that I'm going to be talking about is involved in some of the commercialization work we're doing. So, you know, we're all in this kind of space of genetics where ultimately in order for us to be able to work with our patients, we need to be able to diagnose them accurately. That means, you know, achieving a genetic diagnosis. And for many years now, we've been kind of chasing the promise of genomics to try to, you know, understand majority of heredity, and we're still kind of plateauing at this about 30, 40%, you know, some might say 50% really if you're lucky, diagnostic yield. We know that there's a lot of this information that's buried in our genome. In spite of sequencing our genome, it's hard for us to understand the genetics context. So what I'm going to do over the next 25 minutes or so is try to give you a bit of a general idea of the epi-signatures and DNA methylation. And just out of my own curiosity, by the show of hands, how many of you have heard the term epi-signature? Wow, that's great. Wow, this, I keep asking this question over the last few years, and I guess, you know, there's a bit more knowledge and understanding. So I'm going to start, before I get into it, I'm going to start by just simply defining epigenetics. So it's a study of heritable changes in gene expression or cellular phenotype caused by mechanisms other than changes in underlying DNA sequence. Now, I did underline this other than, and I question it. This is a classical definition. If this definition is correct, the rest of my talk is not about epigenetics, which clearly you will see it is. So we need to think about redefining what actually, epigenetics definition is. The one key thing that's important to mention is that unlike genetic changes, epigenetic changes may be environmental responsive or plastic and reversible. Okay, so epigenetic mechanisms, two of the more commonly studied ones are DNA methylation, histone modifications. DNA methylation takes place at a fifth position of a cytosine ring in coordination between histone modification and DNA methylation. Nucleosome, chromatin, and ultimately chromosomes are assembled. We know it's important to have appropriate chromatin and chromatin regulatory because that's how our genes are turned on and off. And euchromatin or open chromatin tends to be associated with unmethylated DNA, whereas hyper or methylated DNA tends to be associated with the heterochromatin. This is one of the many functions of epigenetics in general. One of the things you have to kind of consider, as we're going to talk about today about utilization of epigenomics in diagnostics, is that epigenetic profiles are very different in all tissues. You know, you can draw blood or get a fibroblast or a buccal swab. You ultimately get the same DNA. If you do DNA sequencing, you don't get the same epigenomic profile. So that becomes very important when you start thinking about strategies or implementing epigenomic testing in clinical setting. A lot of what I'm going to talk to you about deals with data that's generated by microarray technology. We currently use these 850K arrays from Illumina. They're arrays that are designed to measure methylation status, DNA methyl status across 850,000 sites across the genome. Most human genes and CpG islands are covered. We are starting to do some work with sequencing-based epigenomic or DNA methylation technologies, but for the purpose of the talk today, we're going to really focus on a lot of work we've done with the microarrays. So when we started using genome-wide methylation microarrays, I run a diagnostic program in Canada in one of the labs, and we asked a very simple question: can we use the microarray to replace a bunch of tests that we currently use as targeted tests for imprinting disorders? As you all know, historically, Prader-Willi, Angelman, Becker-Wiedemann, various different UPD syndromes, we used to test with targeted assays, so we still do it to an extent, but it was very clear that we could very easily use these methylation microarrays. And just to kind of orient you here, each line is a patient. The purple lines are reference controls that are hemimethylated in 15q11.2 region. Prader-Willi individuals, hypermethylated. Angelmans are unmethylated. So you don't need fancy bioinformatics. You just need to visualize the region, and you can basically screen for this. Similar for Beck-Wiedemann, as you can see, you can pick up different levels of mosaicism. Multiple imprinting defects are indicative of a UPD. So the other really interesting thing that came up when we started using these global methylation microarrays, you can also pick up some of these trinucleotide repeat disorders that cause hypermethylation of promoter. A good example is FMR1, Fragile X. So trinucleotide repeat disorder in males causes hypermethylation of a promoter. After 200 repeats have been reached, as you can see very clearly, all males are easily distinguishable on these methylation arrays, and we can actually pick up mosaics down to about 10% as well. But that's not what I'm here to tell you. So, I mean, yes, we knew we could use genome-wide arrays to try to pick up imprinting disorders. But then, as we were doing some of the early work, we noticed something really interesting. I'm just showing you an example here. You can see a bunch of lines here. There's a couple of hundred patients. Each line is a patient, and showing 3 different genomic regions. And you can see a couple of lines deviate. They're from individual patients. One of the patients happened to be a patient that had an NSD1 mutation, Sotos syndrome. The other was a newborn with dysmorphism, large tongue, and a few other kind of clinical features that resembled possibly Sotos syndrome. We went back to the physician and asked them questions about this patient. They went on and ordered the testing, and lo and behold, found an NSD1 mutation. So it was the first evidence that we could actually use epigenomic profile, very rudimentary at that time, to try to identify a patient with an underpinning genetic defect. So that's where the term epi-signature was derived. And the way that we defined it is a recurrent epigenomic pattern associated with a common genetic or environmental etiology in a specific patient population. I use word environmental because of some of the work I'm going to show you later as well. It's not just genetic. So how do we derive an epi-signature? Normally we start with a cohort of patients. 5 to 10 patients with a common genetic etiology are required. Peripheral blood, we draw peripheral blood. Some disorders have less robust, some have more robust methylation profiles, so in some cases we actually require a lot more samples. And some conditions that we're just not able to at this point pick up these methylation signatures. Epi-signature is a subset methylation profile from the global methylation profile, and it's derived by use of sophisticated bioinformatics and analytics procedures involving machine learning. And what you ultimately end up with is these kind of outputs. This is a Euclidean clustering plot, this is an MDS plot, and this is what we call an MVP plot, support vector machine-derived binary classifier, that tells us that this particular patient has, in this case, it's Kabuki syndrome methylation signature and not any one of the other ones down on the bottom that we mapped so far. Combination of these factors then ultimately allow us to identify cases with specific epi-signatures that are specific to specific disorders. So over the years, our goal had been to really map as many of these signatures as we can. So that's a large international effort I'll tell you a bit more about that currently is ongoing and continues. But we know now that there are well over 100 epi-signatures mapped and defined, and publications in this space keep kind of growing. But certainly it's going beyond conditions that we would have classically considered chromatinopathies, so the disorders of chromatin regulatory genes. We're picking them up in lysosomal disorders, in neuromuscular disorders, in environmental conditions, in a fairly actually wide variety, in epilepsy-related syndromic conditions, and so on. So next few slides, I'll tell you about some of the kind of common general principles when we think about epi-signatures. One of these is methylation variant pathogenicity score. It's a score that is derived based on machine learning, based on methylation profiles of patients, that is specific for a specific genetic condition. So you can have an MVP score for Kabuki syndrome, for Sotos syndrome, and so on and so forth. And then that allows you to really simultaneously screen for many of these different conditions using the same assay. genome-wide methylation rate. So you generate methylation data and you screen for many of these different MVP scores. And what this shows here is individuals with ATRX score very high for ATRX and not for any one of the other syndromes that we published on one of our earlier versions of the classifier. And this kind of goes for other disorders here as well. So that's the one key. The other one is that these epi-signatures are not always gene specific alone, right? So we know that genes work as part of protein complexes and networks. We've now defined many different signatures associated with protein complexes. A good example here is the SWE/SNF remodeling or BAF complex methylation profile. As you can see, individuals with mutations in a number of different genes causing a number of different syndromes that are part of the SWE/SNF remodeling complex have a common DNA methylation profile relative to controls. Now, as we've expanded the database and tested more and more patients, we're now able to identify substructure, including specific gene methylation subsignatures. So we can tell you whether or not patient has a buffopathy and also whether or not patient has Coffin-Siris type 1, for example. The opposite is also true. We now are mapping signatures that are sub-gene specific down to single domains, sometimes even single amino acids. There are signatures sometimes that are associated with a truncating loss of function mutations and a different signature associated with a gain of function mutation in the same gene. One of the things that this technology has been really useful for is to help define new syndrome. As we identify new genetic conditions, we assemble patient cohorts, try to match them based on a clinical phenotype. One really good, strong functional evidence that they have common etiology is that they share a methylation profile. So we've been publishing a lot of new syndromes as a result of, at least in part, having a common DNA methylation signature. We're able to learn new things about all disorders. One thing that's really interesting is microdeletion duplication syndromes. One of your colleagues here, Barb from Greenwood, has been really instrumental in trying to put together international collaboration in this space. But essentially, we're trying to map methylation signatures associated with different microdeletion duplication syndromes. You might ask yourself why, but there could be many different reasons why they will be informative. There are examples of conditions where we're able to refine a methylation signature down to a specific causative gene that may be culprit and part of the clinical phenotype. As an example, in Phelan-McDermid syndrome, where we'd actually associate methylation signature with a subtype metabolomic profile within the patients of Phelan-McDermid syndrome. And then, as I said, there are many other layers of information that we can kind of achieve by looking beyond simply genetics in this case. One of the things that we're really interested in is penetrance. There are CNV conditions where a healthy mother transmits a deletion or a duplication to a child, and the child's affected, and healthy mother isn't. Can we use epigenomics to try to kind of infer, who may become symptomatic, who may not? Why this technology became quite interesting in a clinical setting is because of its ability as a strong functional evidence to reclassify a variants of unknown clinical significance. As an example of a cohort here where we tested a number of patients, and ultimately, based on the best genetic interpretation, many of them had VUSs as a result of epi-sign analysis. We're simply be able to reclassify them as either having or not having a DNA methylation profile and allowing us to move the VUSs from VUS to likely pathogenic in the cases that they do have the methylation profile. I mentioned this earlier, so this is not an individual test. As we add more and more of these signatures, when we run a methylation profiling, we're able to screen simultaneously for many different disorders. What happens when we do that is we know clinical conditions are oftentimes with similar phenotypes and overlapping phenotypes. We might get patients with suspicion of having a specific genetic disorder actually come up with an alternate or different DNA methylation profile. So as an example of, and I'm just using this top panel up here, so number of patients where there was a suspected CHARGE syndrome. 4 of them actually came up positive, number of them were negative for the different classifiers. They were on one of these older versions of the methylation classifier. And one patient actually didn't come up positive for either of those two groups, but rather for a different group. In this case, patient had an ADMP-related signature, and when we went back to the exome data, this patient was missed, had actually complicated variant that was missed by exomes and ultimately was able to be picked up. There are dozens and dozens and dozens of such patients that we've identified. So they get submitted for testing for a particular condition. With suspicion, if you do the full screen, you oftentimes pick up an alternate condition based on a methylation profile that then ultimately allows for targeted genetic assessment. And oftentimes, actually, as a matter of fact, and I should switch to the next slide, when you do things like genome sequencing, comprehensive assessment, you may actually generate the data, but the analytical filters may not allow you to actually identify these genetic variants. As any of you know that do exome or genome sequencing, it's an example of a cohort where we applied methylation profiling, identified the number of patients with methylation signatures that then went back to try to ultimately identify specific genetic variants. We published that a little while ago. So and we're seeing this kind of all the time now. Oftentimes, it's allowing us to pick up variants that are deep intronic variants that require things like mRNA assessment to demonstrate kind of exon skipping. Sometimes they're complex variants that are not being picked up sufficiently by sequencing because of the allele bias on the amplification. Anyone of you that runs on the lab side knows that if you have a complex variant, the other allele might be overrepresented and enriched, and this one will be just filtered out by the software algorithms and so on. So we moved ahead and developed the initial utilization of this and as a clinical test. One of the first labs is Greenwood Genetics Lab here and a lab in Europe in Amsterdam. Since then, there are a few other labs that have come on board, and the network is actively expanding. But this was to really try to kind of move from science and research-based assay into clinical testing and move it through kind of health regulatory through the various different networks. And as the test expands, of course, the list of the genes expands. This is version 3 of the classifier. We're now on version 4, I think about 80 different assays, and in September we're going to be launching version 5. It's going to have over 100 different genetic diseases. What I'm trying to say is that we now have over 100 different genetic diseases where vast majority of VUSs can be reclassified. So there's no reason to have a VUS in many of these genetic disorders. Any one of you that does this testing in patients, oftentimes it requires family follow-up, identifying alternate families of the same variant, developing functional assays. Many of these patients go on for years, sometimes decades, without having the resolution, which creates complications in family planning, access to services, et cetera, et cetera. So when we started doing this testing clinically, we published the first paper of clinical utilization. Looked at first couple of hundred individuals we tested between the labs. They were offered the testing and demonstrated about 35% patients were we able to reclassify variants to likely pathogenic. So clearly significant diagnostic yield and utility. So we're just summarizing a paper that's looking over about 2.5 years of testing up until the end of, I think, 3 years up until the end of last year, with about 3,000 patients tested, and we're seeing similar diagnostic yield. About 30% patients get a positive result. And oftentimes the patients with VUSs that are being reclassified. We're also testing patients where there's no VUS or genetic variant identified. It's about 10% yield in those patients. Oftentimes they're submitted for testing because of strong clinical suspicion in spite of not having a genetic variant. It's just based kind of volume increases. So certainly there's an adoption for the test. So one of the challenges with developing new tests and new testing technology, I need to develop kind of health system impact evidence. One of the big studies we're running in Canada right now is Episignatures, where we're testing about 2,000 patients, either as tier 1 or tier 2. So as part of the initial diagnosis or post-extensive genomic testing. And we're seeing substantial impact diagnostic yield, fairly broad criteria for testing, about 10 to 15% positive. We're trying to run it as though it's a clinical test in Canada, about 22-day turnaround time. There are a number of other health system studies that are ongoing. We're doing some work with with a group in Australia focused specifically on syndromic epilepsies. Recently, we've completed a study through with Manchester Lab through the National Health System in UK, and as of July 1, Episignatures now on the test registry, so it's available for testing clinically in UK and paid for. We're doing another large study focused on mitochondrial metabolic disorders, and I'll tell you a little bit about this study that I'm really excited about. We just learned that we're going to be funded about. It's under embargo, so I can't tell you the funding agency, but this is a very large-scale study, looking at international implementation of this technology. So we do a lot of research discovery. At the moment, there are well over 100 different institutions in 30 plus countries that are collaborating on various different aspects of epi-sign research. Our goal is to test about 1,000 syndromes within the next 5 years. We're actively recruiting for about over 500 right now. And as I mentioned here, and this is kind of a thank you slide because it's really just highlighting some of the key collaborators. There may be quite a few of you in the room here. The network's expanded over the year, and I really thank you. It does take a village to do this type of work. Rare disorders are very difficult to study by any one lab individually. So what do we see for Episign kind of going forward? So as I mentioned, we have over 100 disorders that we can actually resolve USs for and screen patients. We're now looking at the ability to detect teratogenic exposures. So we know that some of the patients we think have genetic conditions actually have prenatal teratogenic exposures, and unlike genetics, epigenetics can be responsive to environment. I'll tell you a bit about epivariants and the metascore. Couple of different examples. This is a study we're just about to submit. So women that have epilepsy seizures when they're pregnant will often take valproate to control these seizures to keep their pregnancies. Estimate about 5 to 8% of the kids are affected by what's called the fetal valproate syndrome. It's really hard to determine for sure if that's the cause. Sometimes there's genetic phenocopies and so on. So we now have a very distinct DNA methylation signature associated with fetal valproate syndrome. And this was a really interesting cohort to go after because valproate, as you know, affects histone acetylation, right? So we thought that was kind of an obvious one to go after. And now we have an ability to detect what is genetic phenocopy, essentially from the environmental exposure, by a molecular assay that's highly sensitive and specific. There's not really a good precedent for this, so we're still trying to figure out what's the most optimal way to offer this type of testing, probably not just as routine screening. We're seeing some really exciting data with fetal alcohol syndrome. That study that's ongoing, we've seen very good evidence of a DNA methylation signal where we're now able to reproduce it on a large validation cohort. This study is a little bit off. Probably by the end of this year, we're going to look at submitting this study. But again, another very important area of environmental exposures, things like diabetic embryopathy, and a few other conditions that we're kind of focused on. Thalidomide exposure is another good example. We're currently collecting cohorts to try to see if we can map these methylation signatures. So now that we've developed these large data sets, we can start looking at these single events, not genome-wide methylation changes, but punctured single events of hypermethylation of promoters in genes that are OMIM genes. So autosomal dominant gene can be, turned off by a mutation, or presumably can be turned off by hypermethylation promoter. Oftentimes, these promoter-specific events of hypermethylation are associated with in-cis genetic variation that we currently don't have very good ability to understand. So if a genetic variant affects how CpG island is methylated, this might be another way. This is an example of a kid, for example, with hypermethylation of VRK2 promoter. This child had idiopathic seizure. VRK2 gene has recently been recognized as an OMIM gene, autosomal dominant gene for epilepsy, right? So we're looking to actually expand into using this technology for this purpose in the coming little while. So, Episign Metascore. So a lot of this work is fairly complicated. A lot of it requires generating data. Data is being generated around the globe. My lab does a lot of the data analytics that go into this. So we're trying to now actually to create software that's going to automate this process and enable every lab to be able to do this analysis independently. So we're trying to combine various different ways or different levels of analysis, things like MVP score, hierarchical clustering, MDS, and so on, and in a very simplified term, turn them into numerical values that could simply be validated empirically. So this is an example of what I'm talking about. So each one of these dots is a patient affected by one of these epi-signature conditions. So we're creating numerical way to capture various different layers of information and ultimately embed them into a single, visualizable kind of output that doesn't require bioinformaticists looking at, t-SNE MDS plots and trying to figure out what a patient fits to patient cohort otherwise. And the reason why we have to do this is because of this project that I'm really excited about. I just mentioned this. So we partnered with an industry partner to build a new microarray. So we know now that one of the first tests that's done in patients with rare diseases is a cytogenetic microarray. We estimate about 1 million plus kids in Europe and North America get a cytogenetic microarray still as a first-year test with about 14% diagnostic yield. We're looking to embed DNA methylation profiling into the cytogenetic microarray test. And the idea would be that as we develop this tech, We would then be able to screen for copy number alterations, for things like imprinting disorders and various trinucleotide repeat disorders that have a methylation defect, and of course DNA methylation signatures simultaneously. So the idea is that there would not be a net new cost to the system, it would simply be generated with every patient that actually gets the test, which I think, and we need to get to a point where we're able to analyze more than thousands, perhaps thousands or hundreds of thousands of patients simultaneously. And that's why we're looking to actually get the software into the hands of the labs to be able to do it independently. I don't have a lot of time, but I will tell you there's this whole other angle to this data and this study. So I've showed you the diagnostic utility, but there's functional implications. So we know that these genome-wide methylation profiles overlap. We know that different conditions are more similar and more dissimilar than each other based on the overall methylation overlap. And these are some of the plots that show overlapping methylation changes in different conditions. Why we think that's interesting is because it's now allowing us to build maps of epigenetically similar genetic conditions. And as an example, here's a number of these disorders that show overall hypomethylation of a very common feature, which is either systemic undergrowth or overgrowth. But there's many other layers, so paralogous genes that belong to the same pathways, patients with certain similar clinical features. This is one of the older versions of the published diagram. But as this builds out, it's going to tell us which genetic conditions have similar epigenetics. And why is that important? Because, a lot of these methylation changes are strongly enriched for CpG gene promoters. They are enriched for neurodevelopmental networks, so we don't think they're random. The question is, are there shared methylation signatures across phenotypes or clinical presentations? Can we correlate them to human phenotype ontologies? And then, can we associate in phenotype severity, penetrance, progression, treatment response? One of the really interesting questions in rare disorder space is, well, when we currently develop drugs for rare disorders, we often target a single gene, maybe down to a single amino acid in a single gene, and the drug might only apply to 20 people around the globe. These are very expensive drugs to develop. So can we look at ways to start thinking about common epigenetic pathways as potential targets that would be a bit more broadly applicable to patients with rare diseases? And that's this whole other area that no one's really explored systematically, and we're hoping really to start kind of sinking our teeth into as these data sets expand. And I'll finish it off with a controversial proposition, and that is that genome is not the most unique code of life, in spite of what you've been told. Epigenome is a reflection of the DNA in a spatial environment and temporal developmental stage context is a much more specific and accurate code of life. And a gentleman named Heraclitus has figured this out a long time ago, way before us, and he said, no man ever steps in the same river twice, for it's not the same river and he's not the same man. And with that, I thank you and ask for any questions.